Very good afternoon to all the students of BIPC. Yes. Just a second. So yes, very good afternoon to all the students of BIPC. This is your Prasanna Ma'am from NEET World, and today we are back to discuss the questionnaire of APEAPCET 2025. Okay, APEAPCET 2025 that has happened recently. So yes, this will be the last set of question paper that we are discussing. If you look into each and every question, we are going to recall from which chapter, from what segments the questions are asked, in what patterns. Okay, mostly it is statement based questions, assertion reason questions are only asked, which test the depth of the subject. Okay, and if you look into this question paper, the first question is from living world, diversity of living organisms, living world, wherein this question is asked from binomial nomenclature, which is given by Carolus Linnaeus. Identify the universal rules of nomenclature among the following. He is asking us to identify the universal rules which are given by Carolus Linnaeus. First, biological names are in Latin, but they are written in italics. Okay, they are printed in italics and they are from Latin origin. The first word to start with small letter for genus, wrong. It is not starting with small letter. For genus it starts with capital letter. For species it is with small letter. Okay, and the specific epithet to start with small letter, this is true. Each organism name will have two components. Every organism will have two components. Okay, so identify the universal rules of nomenclature among the following. If we have to identify the universal rules, yes, of course the names are derived from Latin and they are written in italics. It is not starting with genus is starting with capital letter, not with small letter. This is wrong, and C and D, both the statements are true. So with this we have to go opt what are the correct statements with respect to binomial nomenclature, A, C and D. Right ma? Next question. Choose the correct statement among the following. Okay, this is from biological classification, virus concept. Okay, so now here if you see ma, Pasteur given the name virus. Pasteur is one of the pioneer scientists who has worked on the viruses. This statement is true. Lichens are good pollution indicators, that is correct. But now with these examples I would like to tell you how lichen can act as a good pollution indicator, which is important. What is the internal physiology that is happening? Lichen is a composition of algae and fungi. We all know that, right beta? Lichen is a composition of algae and fungi. Okay, algae is photosynthetic in function. Okay, algae is photosynthetic in function. When it is photosynthetic, it has chloroplast and it has chlorophyll pigment inside it, right? It has chlorophyll pigment. What is the structure of chlorophyll? Chlorophyll has tetrapyrrole ring and a phytol chain. Okay, chlorophyll has tetrapyrrole ring and a phytol chain. So this tetrapyrrole ring is connected to magnesium at the center. Okay, this is the pyrrole ring and this is phytol chain. Clear beta? So chlorophyll pigment whose structure is having pyrrole ring and phytol chain, and these are carbon molecules are connected to magnesium at the center. Whenever this lichen is in close proximity with pollution agents, like what? Nitrogen might be, or sulfur dioxide. Mostly it detects sulfur pollution, sulfur dioxide. Magnesium will lose its structural, it will break its structural integrity, and magnesium will bind with sulfate, forming magnesium sulfate and magnesium nitrate. That is how the chlorophyll structure get dismantled, photosynthetic activity get dismantled, and algae will not provide food to fungi. Fungi structure will be collapsed. That is how lichens cannot survive in the polluted areas. Clear ma? Now you have understood how lichens are good pollution indicators, and most widely it will indicate sulfate pollution. Next, 3 domain groups were originated from progenote. Clear, correct. Okay, 3 domain system of classification which is given by which scientist? Carl Woese. Carl Woese gave 3 domain 6 kingdom system of classification. Those 3 domains have a common ancestor. The common ancestor is what? Progenote. Okay. Chrysophytes belong to monerans. Wrong. Chrysophytes belong to unicellular eukaryotic kingdom, that is Protista. Okay, what is it beta? Protista. So with this we have decoded this question and we have linked up with few concepts also. We have to identify the correct statements from it. What are the correct statements? 1, 2 and 3 are correct. Chrysophytes doesn't belong to Monera, they belong to Protista. Clear, right ma? Next question you see. This is from Science of Botany, okay, wherein the concept and scientist names is given. This is from the chapter Science of Botany. Okay. So here you see C4 photosynthesis has the scientist V.S. Ramadas has contributed in C4 cycle, and plant tissue culture. Plant tissue culture is given by whom? Plant tissue culture Comrade, and genetic nature of RNA is given by Simcura, and sexual reproduction of flowering plants by Camerarius. Once you look into the scientist properly, yes, genetic nature of RNA, plant tissue culture by Simcura and genetic nature of RNA by Conrad. Sexual reproduction of flowering plants is by Camerarius. This is correct only. Just correct this, that is plant tissue culture scientist is Simcura and genetic nature of RNA is from Conrad. Okay. So this is just memory based. Here you see this is from plant kingdom. Okay. Again let us go into the concept. So this question is from plant kingdom beta. Here also they are asking us to identify the correct statements. Okay. Diplohaplontic life cycle can be seen in Bryophyta. No. In Bryophyta the dominant plant body is what? Gametophyte. Okay. Gametophyte is the dominant plant body. What? Sporophyte is dependent on gametophyte. Sporophyte is dependent on gametophyte, and the major plant body is gametophytic plant body. That is why the life cycle here in bryophytes is haplodiplontic life cycle. Haplodiplontic life cycle, not diplohaplontic. In volvox, meiosis and zygote results in haploid spores. True. The only unique feature of algae is what? Zygotic meiosis. Okay, there is no embryo formation in algae or Thallophyta. Zygote undergoes meiosis. Okay, zygote, after undergoing zygotic meiosis, it releases haploid spores. Those haploid spores landing onto the suitable substratum, they grows into the new plant. Next, Cycas has siphonogamous oogamy. True. Cycas will have siphonogamous oogamy. Next, mycorrhizal roots can be seen in the gymnosperm. Cycas. Wrong. Cycas belongs to gymnosperms, right? So it is having siphonogamous oogamy. Up till bryophytes and pteridophytes, they are having what? Zooidogamous oogamy. Gymnosperms and angiosperms will show siphonogamous oogamy. Just look into that beta. So mycorrhizal roots are seen in Cycas roots. Wrong. Mycorrhiza association are with pinus roots. Okay, mycorrhizal association is with pinus roots. With respect to Cycas roots, the symbiotic association, they are symbiotically associated with blue-green algae or cyanobacteria. Okay, blue-green algae or cyanobacteria. They are symbiotically associated with BGA or cyanobacteria. That is why these roots are called as coralloid roots. Coralloid roots. Okay, the main purpose of this symbiotic association is nitrogen fixation. Okay, it is nitrogen fixation. Clear beta? Okay, so here we have to go with correct statements. Those are Statement 2, which undergoes zygotic meiosis, and 3, as cycas as having siphonogamous oogame. These two statements are true. Next question, if we have to look into, this is from Morphology of Flowering Plants, wherein Nepenthes, which is an insectivorous plant, okay, Nepenthes is an insectivorous plant, wherein upper petiole is modified to tendril, lower petiole is modified to a phyllodes, and the leaf apex is modified to lid. Leaf apex is modified to lid, and picture is modified. Picture is modification of what? Leaf lamina. Fine. And here stipules are present, large stipules will be present. So, this leaf apex, which is acting like lid, they will trap the insect, okay, they will trap the insect and they will get the nutrition from insects. That is why they are insectivorous plants. Here what you have to remember, the petiole, upper petiole is modified into tendril, lower petiole is modified to phyllodes, middle lamella is modified to picture. Remember that. So, assertion is true only. Coming to reason, this modification is to trap insects for their nitrogen requirement, nutrient requirement, that is nitrogen. This is true. Here assertion and reason is true, and reason is explaining the assertion. Okay, so here we have to go with the option 1. Clear ma? Next question. Any doubts anywhere, you can message me in the chat box, but I will answer your doubts. So, this is also from Morphology of Flowering Plants with respect to different types of flowers. Acropetal sessile flowers. Acropetal sessile flowers, they belong to what? Acheranthus. Okay. Next, 3-flowered cyme. It is of cymulose, specialized inflorescence. Panicle belongs to compound racemose. Panicle is the type of compound racemose inflorescence. Unisexual flowers with irregular arrangement. Unisexual flowers of irregular arrangement, this belongs to a specialized inflorescence, that is head or head type of inflorescence, or you will also call it as hypanthodium. Okay, this you will also call it as hypanthodium. Now, what are the options? Like A is for 2 Acheranthus, acropetal sessile flowers, 3-flowered cyme for cymulose. Panicle is a type of compound inflorescence, compound racemose inflorescence. Unisexual flowers with irregular arrangement belongs to the specialized flower, that is hypanthodium. Next, this is again assertion reason question. As I told you, most of the questions are statement based and assertion reason only. External fertilization occurs in flower, that is wrong. First you should have clear idea about external fertilization and internal fertilization. External fertilization is fusion of egg and sperm outside the body of, whether it is plant body or animal body. Outside the body of organism, fusion happens, then it is external fertilization. Fusion of male gamete and female gamete inside the plant body or inside the organism is called as internal fertilization. We all know in angiosperms double fertilization takes place, double fertilization, where you will also call it as triple fusion, double fertilization or triple fusion, that happens inside the female gametophyte, okay, inside the female gametophyte. What is the female gametophyte of angiosperm? Embryo sac, okay, it happens inside the embryo sac. Clear beta? Next, in internal fertilization, organism's egg is formed inside the female body, that is true. So, here assertion is false and reason is true. We have to go with the option 4, assertion is false and reason is true. Next question, look into it. This is from sexual reproduction of flowering plants. Okay. So, here from this question, it is asking about the anther wall layers or pollen grain wall layers. Exine is the outer wall of the pollen grain. Exine exhibits array of patterns of designs. That is true. Okay. Pollen grain has 2 wall layers, exine and intine. Exine wall is made up of what? Sporopollenin. Okay, exine wall is made up of sporopollenin. Intine wall is made up of what? Cellulose and pectin. Cellulose and pectin. And mostly in 60% of angiosperms, in what celled stage pollen grains are liberated? Two celled stage. One is large vegetative cell and one is small generative cell. Clear, ma? So now if you see, exine exhibits array of patterns of designs, true. Tapetum layers of microsporangium will be useful for dehiscence, wrong. Let us quickly recall the 4 wall layers of anther. So here this is the structure of anther. Anther is having 4 wall layers, clear, ma? First is epithecium, next is endothecium, next 2 to 3 layers of middle layers. First is epithecium, second layer is endothecium, and then 2 to 3 layers of middle layers, 2 to 3 middle layers. Clear? Epithecium, endothecium, 2 to 3 middle layers, all this together are of protective function. They are of protective in function. Next, innermost layer is what? Tapetum. Tapetum is nutritive for sporogenous tissues. So at the center you have sporogenous tissue, right? So tapetum provides the nutrients for the sporogenous tissue. It is not protective in function. Clear, ma? Next, the intine wall of pollen grain is made up of cellulose and pectin. Correct, right? Intine wall of pollen grain is made up of cellulose and pectin. Next question, by wall formation 8-celled embryo sac is developed. Wrong. After 1 meiosis and 3 sequential mitotic divisions from megaspore mother cell, 7-celled 8 nucleated condition is formed. Okay, what is it? 7-celled 8 nucleated condition. 7-celled 8 nucleated condition is formed, that is in the embryo sac. Okay, no, it is not 8-celled embryo sac, that is 7-celled 8 nucleated. So now here you see, ma, what are the correct statements he is asking. 1 and 3 are correct. Just recalling the concept so that you will be revised with the concept. Okay. So this is again from morphology of flowering plants, just from the examples of the families. Okay. So blue dye it belongs to, okay, blue dye is given by what? Indigofera tinctoria, this is of Fabaceae. Next, Sarsaparilla, so this is from Smilax. Next, yellow dye from Butea monosperma. Next, chemical mutagen is from the plant Colchicum and attenuata. Okay, Colchicum is belonging to the family Solanaceae. So just by looking into this two, immediately this will strike to you, because those are from Fabaceae and Solanaceae. Those are common families for NEET and this, no. So this will be registered in your mind. So you can identify the two statements and you can eliminate the other options also. In this way also you can solve the question, match the following questions. The beauty of match the following question is you can easily decode, okay, you can easily decode and sort it out, fine. Next question, ma. The longest cell, this is from Cell the Unit of Life. The longest cells are nerve cells, okay, which are branched and long. Smaller DNA in bacteria called as genetic DNA. We all know bacteria has one genomic DNA, which is the long DNA, which is at the nucleoid, and one small DNA, this is called as plasmid DNA or non-genomic DNA. It has some particular functions like giving specific phenotypic character to the bacteria, providing antibiotic resistance and promoting conjugation, right. So this helps in that 3 particular features, it is not with respect to genomic DNA. This is wrong. Next, bacterial flagellum has filament, hook and basal body, correct. Next, in bacteria, flagella are used to attach for rock. With respect to prokaryotic cell or bacteria, it has both motile and non-motile structures, beta. It has both motile and non-motile structures. The only motile structure you have is flagella. Okay, flagella is the only motile structure you have. Non-motile structures, if you have to look into, it is pilli and fimbriae. Okay, it is pilli and fimbriae. How you can describe pilli? These are tubular extensions of the cell wall and they help the cell to attach to the substratum, or they help in cell to cell connection. Fimbriae, this you can describe as bristle like fibers sprouting out of the bacterial cell. They purely help in attachment of the bacteria to the host cell or substratum. Pilli has additional function of supporting conjugation, which is a sexual recombination, by forming what? Sex pillars between two bacteria. Okay, it forms sex pillars between two bacteria, the one which is of having F strain, that is F plus, and one with F minus strain. Okay. So, this is about motile and non-motile structures. Flagella is not a non-motile structure. So, this is wrong. What are the correct statements? Correct statements include 1 and 3. Clear ma? Next question. Yeah, this question if you see, choose the correct statement among the following. So, here this question is from again cell the unit of life only. Proteins synthesized by ribosomes are modified in cisternae, are released from the transphase. So, this is from endomembrane system. Okay, this is from what segment of the chapter ma? Endomembrane system. Endomembrane system. So, this if you have to consider endomembrane system, proteins are synthesized by ribosomes, yes, from rough endoplasmic reticulum of ribosomes. Where is endoplasmic reticulum present? These are the extensions of nuclear envelope, and over the endoplasmic reticulum, if ribosomes are present, you call it as what? Rough endoplasmic reticulum. Okay. Facing the rough endoplasmic reticulum, what cisternae are present? Cis phase of Golgi. Okay, it is cis phase of Golgi, or convex phase of Golgi is facing the endoplasmic reticulum, and towards the cytoplasm, transphase of Golgi is present. Okay, towards the cytoplasm, what is present beta? Transphase of Golgi. This is concave or transphase. This is cis or forming phase. Cis or forming phase, this is the trans or concave phase, or maturing phase also we call it as. So, taking the naive proteins, packing happens inside the cisternae and they will release out in the form of vesicles, those are lysosomes, or they will also release out glycolipids and glycoproteins. So, this statement is true. Primary wall is formed, first formed layer of the cell. Primary wall layer is not the first formed layer, middle lamella is the first formed layer in the plant cell. Okay, what layer beta? Middle lamella. Middle lamella is the first formed layer in the plant cell. Let us quickly recall the cell division of a plant cell. In the telophase stage, when two nuclei are towards the opposite pole, what happens is, first in between the two nuclei or two cells, first cell plate is formed. This you will call it as phragmoplast. This phragmoplast is being synthesized from Golgi bodies. Okay, this is being synthesized from Golgi bodies, and this phragmoplast moving towards the peripheral sites. This is centrifugal pattern of cell wall formation. Then it leads to the formation of middle lamella. Okay, phragmoplast forming cell plate, this forms middle lamella. So, in between two cells, what is the first formed layer? Middle lamella is the first formed layer. Next, inner to the middle lamella, primary cell wall is formed. Okay, inner to the middle lamella, what is formed ma? Primary cell wall is formed. Inner to the primary cell wall, secondary cell wall is formed. Secondary cell wall is close to the cell membrane. Primary cell wall is close to the middle lamella, and middle lamella is the first formed wall layer in the plant cell. Remember that. Next, in potato, aleuroplast will store carbohydrates. Wrong. Alluroplast will store what? Proteins. It is not storing carbohydrates. Next, in the presence of magnesium, two subunits of ribosomes are associated with each other. True. If you look into the larger subunit and smaller subunit, okay, larger subunit and smaller subunit. This larger subunit and smaller subunit are associated with each other by magnesium ion concentration. Okay, they are associated with each other by what ion concentration? Magnesium ion concentration. More the amount of magnesium, it is connecting both the subunits, larger subunit and smaller subunit. Less the amount of magnesium, okay, the association will differ. It is not that strong. Okay, so magnesium is deciding the association between larger subunit and smaller subunit of ribosome. So yes, this is correct statement. Now in the question he has asked us to identify the correct statement. Then we have to go with the option A and D. These two are correct, these two are wrong. I have explained you completely why those are wrong statements. And you look into this question, beta. Choose the incorrect statement among the following. Incorrect statement he is asking. Cellulose does not contain complex helices. Cellulose will not contain complex helices, that is true. Okay, it is made up of homopolymer. Cellulose is a homopolymer of glucose unit. So yes, cellulose is not a complex helices. In DNA, one full turn of helical will be 3.6 angstroms. Wrong. One full turn or one pitch or one full turn is 3.4 angstroms, not 3.6, it is 3.4 angstroms. Next, abrin is a toxin secondary metabolite, that is true. Okay, this is from the table of secondary metabolites from the chapter biomolecules. Okay, by grinding tissues in perchloric acid, acid soluble fraction can be obtained. No, acid insoluble fraction which is having biomacromolecules can be obtained. Okay, so here what are the correct statements? We have to go with 1A and C. Next question. Next question. So yes, this is from cell cycle and cell division. Okay, this question is from cell cycle and cell division. Match the following question, look into it carefully. Recombination nodules are formed at the time of crossing over. Okay, it is formed at the time of crossing over. During what stage? Pachytene stage. Okay, it is formed during Pachytene stage. And recombination or crossing over is an enzyme mediated process. What is the enzyme that is mediating here? Recombinase. Recombinase is a enzyme. So recombination nodules are mediating what? Crossing over during the Pachytene. Next, homologous chromosomes. Homologous chromosomes are bivalent condition that is formed during which substage of prophase 1? It is formed during zygotine of prophase 1. Next, interkinesis is the stage between two meiotic divisions. Here in B he is asking you homologous chromosomes separate. Okay, in meiosis anaphase 1. Before anaphase 1 what happens? As bivalent condition is formed in zygotine of prophase 1. In metaphase 1, look here carefully ma. In metaphase 1, two chromosomes are aligned. Okay, two metaphasic plates are formed. So that is why you can observe bimetaphasic plate during metaphase 1. Okay, what do you see? Bimetaphasic plate during metaphase 1 where asters are at the opposite poles giving out the spindle fibers. Spindle fibers will attach to kinetochores of the centromere. Okay, so now this chromosomes are forming what plate? Bimetaphasic plate. Here the chromosomes which are towards this pole will move towards the opposite side. This chromosomes will move towards the lower pole. Okay. So here there is no centromere splitting. There is no centromere splitting in anaphase 1. Remember that, most important statement. There is no centromere splitting. Centromere splitting in anaphase 1. Okay, there is no centromere splitting in anaphase 1. Homologous chromosomes will separate. So this is of option 1. Interkinesis. Interkinesis is the stage between two meiotic divisions. Interkinesis is the stage between meiosis 1 and meiosis 2. Next, splitting of centromere. Centromere splitting will happen during anaphase 2. Okay, in anaphase 2 centromere splitting happens. In anaphase of mitosis and in anaphase 2 of meiosis, centromere splitting occurs. Clear beta? So now this is how you can decode this option. Fine. The activities and phases are given out here. Next question. This is from anatomy of flowering plants. So if you look into the question, identify wrongly matched one he is asking you. Bicollateral vascular bundles. Bicollateral vascular bundles means towards the opposite sides you will have a phloem and at the center xylem will be. This is the bicollateral form of arrangement. Bicollateral you will see in dicots. Best example is solanum. Correctly identify wrongly matched. This is the correctly matched one. Second question, if you have to see, gymnosperms. Gymnosperms will have albuminous cells. Okay, gymnosperms are having albuminous cells and sieve cells. They do not have companion cells and sieve tube cells like how you see in angiosperms. Gymnosperms will just have albuminous cells and sieve cells. Third, if you see, sclerenchymatous pericycle, it is not observed in monocot stem. Okay, sclerenchymatous pericycle you will see in dicot stem. In monocot stem you will see sclerenchymatous hypodermis. Okay, sclerenchymatous hypodermis. Clear ma? Next question, bulliform cells. Bulliform cells are observed in the epidermis of monocot leaf, that is isobilateral leaf. This is true. So what is the wrongly matched one? That is sclerenchymatous pericycle, which is observed in dicots, not in monocot stem. So option 3 will be your correct answer. Right. Next question. Yes. Choose the correct statements among the following. We have to look into the correct statements. Epidermal cells have more cytoplasm and small vacuoles. Just a second. Yeah. Epidermal cells have more cytoplasm and small vacuoles. Outer walls of guard cells are thick and inner walls are thin. This is wrong statement. Epidermis will not have vacuoles. And outer walls of guard cells, if you have to take the guard cell structure, the outer wall is thin and inner wall is thick. That is why it is easy to move for stomatal opening and closing. So outer walls of the guard cells are thin and inner walls are thick. This is wrong. Presence of phloem in the either sides of the xylem is seen in cucurbita vascular bundles. True. Trichomes of shoot system are multicellular. Correct. Root hairs will be unicellular. Trichomes will be multicellular. Correct. So here the correct statements will be 3 and 4. Clear, right beta? Next question. So this you see, this is from annual rings in dicot stem. One annual ring or 1 year of the plant, you can calculate the year or the age of the plant by annual rings. Under the study dendrochronology, we all know that, right? So one annual ring will have one spring wood and one autumn wood. So it is one ring of spring wood and one ring of autumn wood, together they will form one annual ring. It is from secondary growth of anatomy of flowering plants beta. Next, this question you see, choose the correct statement among the following. Okay, this is from photosynthesis of higher plants from plant physiology. Many desert plants will show CAM cycle, crassulacean acid metabolism, where they have scotoactive stomata. Okay, scotoactive stomata, where stomata opens during night time. Okay, this is correct. Next, plant rotation of the plant in the axis will cause seasonal changes. That is correct statement. Ecology is concerned with changes in the environmental conditions. Wrong. Ecology is concerned with interaction between organisms and the environment. Okay, so this is wrong statement. Next, mechanical tissues are developed in submerged water plants. Wrong, because submerged water plants will not have strong mechanical tissues. They are in close proximity with water itself. They do not have high conducting system also. So this is wrong. The correct statements are 1 and 2. Clear ma? Next. This question you see again assertion reason question from ecosystem. Natural ecosystem will perform fundamental life support services without life would cease. Correct, right? They support the fundamental life system. Without that, the life would cease in the natural ecosystems. That is true. Assertion is true. Reason, the dependence on the ecosystems is poorly understood and they can provide life enhancing services. Correct. Why these are fundamental life supporting services is explained in the reason. That is why A and R are true and R is the correct explanation of A. Assertion. Next question. So this if you see ma, choose the incorrect statement among the following. Incorrect statement he is asking. Evolution of C4 plants system is a strategy of maximizing the availability of carbon dioxide while minimizing the water loss. That is correct. Because C4 plants are what plants? Tropical plants. They have a special feature, special anatomical feature, that is they exhibit Kranz anatomy. Okay, Kranz anatomy of bundle sheath cells. And 2 cycles will participate in C4 plants. C4 cycle in leaf or mesophyll cells, and C3 cycle in bundle sheath cells. C4 cycle in mesophyll cells and C3 cycle in bundle sheath cells. So, in C4 cycle the primary carbon dioxide acceptor is PEP and the enzyme is pepcase. This only binds with carbon dioxide, not with oxygen, unlike the enzyme Rubisco which is there in C3 plants. So this adaptation of Kranz anatomy and primary enzyme for carbon dioxide acceptor, that is pepcase, which is the best adaptation for maximizing the availability of carbon dioxide and minimizing the water loss. So this is correct only. Next. In succulent plants, water potential gradient established due to accumulation of organic acids at the night. True, because of the CAM cycle at night time the carbon dioxide is fixed and it is forming oxaloacetic acid, ascorbic acid, oxalic acid, which are organic acids, and they are stored inside the vacuole. This is correct statement only. During night time, guard cells become flaccid in succulent plants. Wrong, because during night time only stomatal opening takes place in the succulent plants, that is desert plants. It is not becoming flaccid. So this is wrong statement. Let us also look into the last statement of this. Light is necessary for accumulation of potassium ions in the guard cells from subsidiary cells. This is true. This we have learnt clearly in stomatal opening and closing mechanism. Clear ma? So here he asked us to identify incorrect statement and the incorrect statement is this. Next, last question of today's class, which is very simple. This is from transport in plants of plant physiology beta. Shrinkage of protoplast due to diffusion of water from cell into the surroundings. Shrinkage of protoplasm due to diffusion of water. This is what we have learned this under plasmolysis. When you have placed a grape in water, okay, the protoplasm gets shrinked when it is placed in the water containing some salt, hypertonic solution. The plasmolysis, shrinkage of protoplast occurs inside the cell. Okay, this is explained under what? Plasmolysis. Let us also look into the other 3 processes. What is osmosis? Transfer of water or movement of water through the semi-permeable membrane from high concentration to low concentration. Imbibition, you can see this phenomena at the time of seed germination, that is absorption of water. Guttation is nothing but the water which is evaporated from the margins, specialized margins of the leaf, which are called as hydatodes. Okay, Hydrothodes are the specialized margins over the leaf structure. Through the specialized margins, the water is evaporated in the liquid droplets itself. Water is evaporated as liquid droplets. Liquid droplets. Fine, ma. So, this process is called as guttation. If water is evaporating through vapor, water is evaporating in the form of vapor through stomata, stomatal pore, that is called as transpiration. But if water is evaporating out through the specialized margins, that is hydrothodes, in the liquid droplet form, then that is called as guttation. Guttation happens in the absence of sunlight during night times or during humid times. Okay. So, with this we have discussed the first set of questions of this paper. In the next session we are going to discuss the next set and we are going to finish the APEAPCET papers totally. Okay. Thank you.